And let's share this screen. Perfect. All right, so as I said, we're going to talk about gel electrophoresis. We're also going to talk about RFLP, which stands for restriction fragment length polymorphisms. It's a way that we can study various different sections of DNA, and you can also use RFLPs to distinguish species or show identity or kinship between two species. So we'll talk a little bit about that towards the end. So gel electrophoresis, it's been around for quite a long time. It's been used since roughly the 1960s. I know gel electrophoresis has been used in protein isolation since the 1970s. It's a different type of gel electrophoresis called sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS, PAGE. And the only reason I know that that's been in use since the 1970s is I used to work in the lab of the guy who invented it, and he did it in 1974. So gel electrophoresis is just, in the most simple sense, it's a way that you can separate out fragments, and particularly for our case here, looking at DNA, it's a way to separate out DNA fragments based on their size. You're also separating out the fragments based on their charge as well. And we'll talk a little bit about that. I'll come back around to that and talk about that when we get to the picture of the gel electrophoresis chamber, because it will make a little bit more sense to see a picture when we're talking about separating it out by size and charge. A lot of this started, and at least cloning, doing cloning of genes into another genome started with Paul Berg in the 1970s. Paul Berg was the first person. Here's a picture of him. He's still living. He's in his 90s now. He was awarded the Nobel Prize in Chemistry in 1980 for showing that his lab and he were the first people to show that you could take DNA from one organism and insert it into the DNA of another organism. And the way that they were able to do this was using bacterial DNA, which are called plasmids, and we'll see that in the next slide. But what he did is he showed that you could take a plasmid from a bacterium, the bacterium's DNA, you could cut it with a specialized enzyme, you could insert a gene into that plasmid, and then you could put that plasmid back into the bacterium, and then you could let the bacterium grow, and then you would have multiple copies of that particular gene. The other thing, too, that came along about this time is that with Berg's findings, people were able now to say, wait a minute, we can stick genes in here that are going to have useful protein products. So other researchers kind of around Paul Berg also took this on, took his information and said, one of the most common examples we give in the 1970s is when they would take the gene that was responsible for producing insulin, and they shoved it into a bacterium, and now the bacterium was expressing a gene that was producing insulin protein that they could isolate, and that was the first insulin that was used, medication that was used for people with diabetes. They don't do it that way anymore. They make it pretty much synthetically now, but that was the first real biotechnology application, I guess you could say, is that we were using bacteria to express genes to make insulin. And another question I sometimes get from students right about here is, well, why did they use a bacterium? Why didn't they use something like yeast? Because yeast is a eukaryote. Expressing these genes in eukaryotes can sometimes be harder than just doing it in a prokaryote. There are pluses and minuses to both, and we don't have time to go into those. We would need a whole class in molecular biology to go over all that. But sometimes it is tougher to get a eukaryotic species to take up a gene from some other organism and get it to express properly. So the bacterium, the plasmids are very small. They're not very large pieces of DNA. They're circular. They were just simply easier to deal with. And so they were using this method here, and this method is still used every day, thousands of times across the world in molecular biology labs. I have done I can't even count how many times I've done cloning of genes into bacteria. So what got the ball rolling here, as I mentioned a little bit before, turn that on, is that we figured out that bacteria have circularized DNA. And this is called plasmids. Another thing that got discovered right about the same time was that bacteria, I'm just going to draw a little arrow over here, we also figured out that bacteria could produce these things called restriction enzymes. And I'm just going to call it RE here because spelling it out will take forever. We're going to see these in a slide here in just a second. Bacteria have the ability to make enzymes that will attack viruses. And these viruses are called, these enzymes that attack viruses are called restriction enzymes. And so there were a lot of scientists also at the same time Berg was looking at that you could isolate plasmids and put DNA in it from another organism, that, hey, we could use these restriction enzymes. We found this stuff that will chop up viruses, so we could probably use those enzymes to also cut up DNA in other organisms or from bacteria themselves. And that's exactly what they did. What you do is you take your plasmid, you isolate it from the bacterium. You're going to cut it with a restriction enzyme, and that's going to leave a gap in the circle. In that gap, you can now take a gene from another organism, whatever you're interested in, and you can insert it into that plasmid. And this is just simply called cloning. You're cloning this plasmid so that you can make more copies of that gene, or you can make more copies of the protein that it expresses. You then take that plasmid, it's now called a recombinant plasmid because you've added foreign DNA, you've added DNA from another organism, and you can put the plasmid back into the bacterium. And now you can just grow this bacterium as you would normally grow any bacterium. Normally, one of the more common ones to use is E. coli. E. coli grows very rapidly at about 28 degrees Celsius, and it grows easily in a lab. So this was pretty much the bacterium that everyone used was E. coli, and that was the one my lab used as well. And what's going to happen now is twofold. You can have more copies of your gene expressed every time the bacterium doubles, or you can have the protein expressed that gene is responsible for producing. And so again, for the protein side of things, this was something like insulin. They could produce a medication using this method. But for gene of interest, and a reason this was also important is at this time, also coming along at this time was trying to figure out how to figure out the sequence of DNA. And when you're trying to sequence DNA, especially in the beginning when they were learning how to do this, they needed multiple copies of the gene so that they could have multiple tests to see how effective their sequencing methods were. And so before there were other methods to make copies of DNA, the only way to make copies before PCR came along, and we'll talk a little bit about PCR in just a minute, this was the way that you got more copies of your DNA, is you simply stuck it in a plasmid. You made a recombinant plasmid, you put it back into the E. coli, you grew up the E. coli, and then you isolated that gene back out of that plasmid. So let's go to the next slide so we can see that. So you could take your copies of your gene back out, or you could take the protein that that gene coded for, and now you could use it to do things. Here, an example from wherever this picture came from, they were doing human growth hormone this way. They were using it for insulin, as we've said before, other medications. You can now essentially make medicine this way. Also protein to dissolve blood clots and heart attack therapy were made this way. If you're looking at the gene itself, okay, yeah, now we've got a lot of copies of the gene. We can sequence it better, or we can insert these genes into plants for something like pesticide resistance, Or we can alter the bacteria with these recombinant plasmids to do something like take up and dispose of things like toxic waste, oil spills from ships. So pretty much the ability or the things that you want to do using recombinant plasmids are pretty endless. You can use them to do a lot of things. And this, for about 20 years or so, this was the main way that molecular biology progressed, was using these recombinant plasmids, putting them in bacterium and isolating the products, and then using those products to perform whatever the next step was in your project. So again, as we've said, bacterial restriction enzymes, again, these were discovered, and these are produced by bacteria to cut up DNA. We're going to talk, I think, let me check here. We're going to talk about gene expression in bacteria in chapter 16. So we will come back around and talk a little bit more about restriction enzymes, because it's going to become important when we talk about how bacteria express their genes. So we'll just say that we can use bacterial enzymes to cut DNA molecules at specific DNA sequences. In these sequences where we know we can cut at in the plasmid are called the restriction sites. And so a restriction enzyme is usually going to make a lot of cuts, thus yielding what are called restriction fragments. And the most useful restriction enzymes that have been found will cut DNA, leaving it with a staggered end. And we're going to see this in a few slides here. But it's going to produce fragments with what are called sticky ends. And they're kind of going to leave the DNA looking like this at the very end. Okay, and those are staggered ends, thus you can hook the DNA back together much like puzzle pieces. Okay, so sticky ends can bond with complementary sticky ends of other fragments. And this is how you can insert that gene that you're interested in into that plasmid DNA. You simply just need to cut the plasmid with a restriction enzyme and then use the exact same restriction enzyme to cut up the gene that you want to insert into there, because it's going to leave complementary sticky ends. Okay, and then once you put your DNA in there, your gene from the other organism you want to put into the plasmid, you got to seal it up, because it's just not going to fall into place and everything is good. You have to use an enzyme to kind of make the zipper complete, and that's called DNA ligase. So DNA ligase is simply, it's an enzyme that will seal bonds between DNA, or in this case, seal bonds between restriction fragments. We're going to see DNA ligase again when we talk about DNA replication later. So again, a lot of these terms we're just kind of hitting on briefly here, but we will talk about them in more detail later in the semester. So again, you have your bacterial plasmid, you've isolated it, you know what your restriction site is. You're going to cut it with a restriction enzyme that you know is going to cut there. It's going to leave you, again, with these sticky ends, as we can see here, little overhangs. You cut your gene of interest from another organism with the same restriction enzyme. It then can fall into place. And then you use DNA ligase to come along, and now you have a solid piece of DNA again. And what you've ended up with is a plasmid that has been cut open and now contains your gene of interest in it. And I hate to say it's that easy, but it really is that easy. These are very simple to do. They only take about 30 minutes to do. The longest part is when you put these back into your bacteria and then having to wait on that bacteria to grow. That takes the longest part. However, I digress here. But this is pretty much the way that, again, as I said, molecular biology progressed and genetics progressed over 20 years, was making these recombinant plasmids so that we could express some protein or do some genetic studies. And so, again, this is just a picture here. You can, just showing here that you can add various different genes into plasmids, you're typically not restricted to what type of genes you can add in here. But as time progressed and as we moved towards the middle of the 1980s, this thing called PCR came about. And so PCR it stands for polymerase chain reaction. And again, we will talk about it a little bit more throughout the semester, but it's a way to make copies of DNA very quickly. So as PCR became more readily used and easier to use in a lab, the recombinant plasmid method kind of fell by the wayside. We still use it a lot, but if you can make a copy of your gene a lot faster, you can make billions of copies of your gene in a PCR reaction usually takes about 4 hours versus waiting 24 hours for bacteria to grow. You're simply going to use it because of the timing factor. However, others also used PCR to make lots of copies of their genes of interest so that they could stick back into lots of copies of these plasmids and make recombinant plasmids out of them to make more product of whatever they were looking at. And this was particularly true when they were trying to work out the best way to sequence DNA. So to check recombinant plasmids, you might cut the products again using the same restriction enzyme. And to see whether or not these, if your gene went exactly where you needed it to go, and to see whether or not it inserted properly into the to make that recombinant fragment, you can use gel electrophoresis. You can separate the fragments so that you could tell whether or not, again, your gene of interest was inserted properly into the plasmid. And so this technique, as we see here, uses a gel made of a polymer. It's made out of agarose. It's a type of sugar that's going to separate mixtures of nucleic acids or proteins based on size and charge, mainly on size and charge. And so a gel electrophoresis chamber kind of looks like this. I'm sure, and I hope that most of you have seen pictures like this before. But what you're doing when you're looking at your well, let me start with A up here before going to the bottom one here. So DNA here, when you add your sample to the wells in the gel here, you're always going to load them on the negative side, cathode side. And the reason is that DNA is negatively charged. Remember, DNA is made up of a backbone of phosphate. It's phosphate sugar, but you have a lot of phosphates out there, and those phosphates give DNA an overall negative charge. So when you're putting your DNA sample into the gel, you want to put it on the negative end. Because what you're going to now do after you've loaded all of your samples in the wells here, you're going to put a cap on top of here, and you're going to add an electrical charge to it. Well, since DNA is negatively charged, it's going to move away from that negative cathode, and it's going to move towards the positive anode. And so there's a saying, a very common saying in labs is run to the red. So when you're setting up your gel, you always know to set it up where it's facing the red side, so you don't put your DNA in backwards, because if you put it in the wrong way, the DNA is going to run out of the wells and go that way, and it's just going to run out and you're never going to see it. Believe me, many people have done this. It's a lot easier to screw up than you think. So what you're looking at after you've separated your DNA in the gel electrophoresis chamber, you then can visualize it under a UV light source. And what you're looking at here is to see, well, I know what size my plasmid is. So you know what size that plasmid is right there. But let's say due to the DNA that you added to it, the plasmid is now a little bit bigger. So the plasmid size, the recombinant plasmid size, is going to change based on that DNA that you added in there. And the old adage, and again, I know this is pretty hard to conceptualize just sitting at home looking at this, but the bigger the DNA fragment, the slower it's going to move in your gel. Okay, because the reason it's going to move slower simply is due to physics. It's bigger. And so what you can do is you can now compare, let's say just for this, this fragment here is the plasmid without your gene put in it. And you say, all right, that's about the size of my plasmid. But then you see this band up here, which is a little bit bigger in size, and you go, well, that's probably my plasmid with my DNA added to it. And so you can use this gel just to confirm whether or not your DNA went in and your plasmid is the appropriate size that you've calculated. All right. So again, we're going to dance around gel electrophoresis the rest of the semester. So when appropriate, I will bring it up in our lectures, and we'll talk about it, how it can be applicable to the subject we're talking about at that time. As I mentioned in a previous slide here, this was all well and good for a long time. We can throw a gene that we're interested in into a plasmid, and now we can grow up a lot of bacteria, and now we have a lot of copies of that gene. But we still weren't getting enough copies to make it very useful for trying to figure out the best ways to sequence DNA. And so the savior was polymerase chain reaction, or PCR. And so PCR is just simply a way that you can take a segment of DNA and you can multiply it many, many times over through a 3-step cycle of heating, cooling, and replication. And so it's simply just a chain reaction going through these 3 steps that cause that fragment of DNA that you're interested in studying, and making a copy of it, so that if you're starting with just one fragment of DNA, at the end of an entire PCR reaction, you end up with about 1 billion copies of that fragment. And as I said, this only takes several hours to do. And you're not at the mercy of growing bacteria that may take 24 hours to have a complete growth curve. Okay, so you're exponentially growing your population of DNA molecules. The way that you're able to do this is that you're using heat stable enzymes that aren't affected by the heating process of PCR. And I'm not going to get too much deeper into this, because we are going to talk about DNA replication later in the semester. So we will come back to this and talk about what's going on here in a little bit more detail. But one of these enzymes that is indifferent to high heat is called Thermophilus aquaticus, or TAC. And it's or Thermophilus aquaticus DNA polymerase, TAC, and it's called TAC polymerase. This was discovered by a guy named Thomas Brock. Thomas Brock's still living. He is still a microbiologist, and he's the world's expert in bacteria that can live in hydrothermal vents that can withstand high temperatures. I can't remember their name right offhand. But he had this student that worked in his lab named Kary Mullis. Here's a picture of him over here, who thought that we could use the enzyme from this bacterium to make PCR better, because the enzyme coming from this bacterium that came from boiling water is not going to break down. And so he was correct. And this is why we use this TAC polymerase in PCR, is that heating step in PCR doesn't denature that enzyme. Thus, we can have the DNA replicate without the enzymes going bad, essentially. And again, very oversimplification here, but we will talk about this again later in the semester. And for his work, Kary Mullis was awarded the Nobel Prize in Chemistry in 1993. A lot of controversy surrounds that, but again, we'd only have so much time. I can't get into that story. So, but the key to PCR is using this heat-stable DNA polymerase called TAC polymerase. Also in PCR, you use other little pieces of DNA that are specific to the region that needs to be amplified, or specific to the area that you're interested in amplifying. It also occasionally will incorporate errors into the amplified strands, but you just kind of discard that because you're only going to have maybe 60 errors out of a billion copies, which virtually makes your PCR reaction 99% error free. So for this slide here, I'm just going to use it to show the exponential growth here. In lab, if we were having lab face-to-face, we would be talking about what's happening here with the plant primer, but since we're not using it, we're not going to consider it here. But in this picture is that here's our target sequence of DNA. You're going to heat the DNA hot enough that it's going to cause the DNA strands to separate, and that's where the heat comes in. You then cool it down just slightly so that these little short primers, these little pieces of DNA that are going to help it become parts of the new strands that are being made copies of, can attach to the DNA. And then you heat it up ever slightly, just enough that it's going to be the most efficient temperature for the extension to occur. And so now that since you started with one molecule up here, at the end of cycle 2, you have 4 molecules. The end of cycle 3 is 8. The next is 16, 32, 64, etc., etc., etc. And again, by the end of the PCR run, you end up with about 1 billion copies of that target sequence. Now, PCR can also be used, and it's been very useful in doing things like crime scene analysis. It also has been used in trying to figure out genetic anomalies or diseases that are found in people. And the way that this occurs, and I think I guess I should have said also at the beginning, I am reading your articles. I'm about halfway done with them, so I hope to have those finished this weekend and have grades back for you. I think someone wrote a paper about this, but I'm not 100% sure. But you can also have what are called single nucleotide polymorphisms. These single nucleotide polymorphisms are little aberrations in the DNA that can be found frequently associated with particular inherited disorders. And so you can use PCR to amplify these regions that you suspect that are associated with these regions that are associated with these disorders. And that way researchers can start to find and figure out, well, what is the location of this disease-causing gene? The single nucleotide polymorphism is rarely directly involved with the disease itself. However, it's just shown that a lot of people who have a particular disease tend to have these polymorphisms in particular areas. They're generally going to be found in non-coding regions of DNA, so you never see any type of problem from them because that region of DNA is simply just not going to get coded. But every person, every individual on Earth, including all of you listening out there, if nobody knew who we were, I don't know how many people are in here, 13, well, 12 people are in here, 13 people, yeah. Let's just say they found us out on an island and we're 13 individuals and they don't know what species we belong to. Well, they could take DNA samples from all of us and they could look for single nucleotide polymorphisms. And all of us, all 13, we're all unrelated to each other. We should still have the same single nucleotide polymorphisms, are very, very similar because we all are human species. So every species on Earth has common SNPs that you can use to say, oh, well, this is that species. And so this is also very useful in species discovery, particularly in finding new organisms. When I was in grad school, the person, the research lab across the hall from mine, they were interested in discovering new species of mushrooms, and this is exactly what they did. They looked for SNPs in those mushrooms that they would find to see did they match anything that was already known, and if they didn't, they would further test them so they could say, oh, we found a new species of mushroom now, and this is part of how you can do these. So individuals have unique DNA sequences, and these are called genetic profiles. So you can use this, again, in things like forensics to identify individuals with a pretty high degree of certainty that that person was part of a crime scene. And you can do this by analyzing genetic markers called short tandem repeats as well. So there's two things going on here. Single nucleotide polymorphisms typically are going to be species based. You're going to see very similar SNPs within a species. Short tandem repeats, though, tend to be more unique to the individual. And so the probability that two people who aren't related having the same short tandem repeat sequence is almost impossible. Okay, so the short tandem repeats here, again, these are just variations in the number of repeats in specific DNA sequences. And you can use things like PCR and gel electrophoresis to amplify those STRs and identify them, be whatever length they may be. Oh, and it says it here, the probability that two people who are not identical twins who have the same STR markers is very, very, very small. STR analysis has been used now very recently, and I mean in the last few, here we got the last 7 years, 2013, to exonerate people who've been put in prison for crimes they didn't commit. And I know you could, we're not a political science or a sociology class. There's a lot of other problems with the criminal justice system, but on the genetic side here, we can use these genetic technologies now to show someone's innocence of a crime using these short tandem repeats. And it says here, this factoid here, as of 2013, more than 300 innocent people have been released from prison due to the analysis of these repeats. So what you do is that you look at, well, here's the defendant, or AKA the person that is being accused of the crime. Here is blood from another person's clothes, and here's the blood from the victim. So you can see the blood found on the shirt here, it belonged to the victim. However, we only see that on the jeans here, we only have these two markers here that sort of kind of match, but they really don't. And so if this person here was accused, this defendant here was accused, wait a second, I got this completely backwards here, sorry. I thought this person was innocent. They're not innocent here. The defendant's clothes here has the victim's blood on it. So there's a very good probability here that this person here killed the victim over here. I was trying to have a happy story here, and rather than having a bad story here. But what we see here is that we have the victim's blood on the defendant's clothes. It doesn't match his or her blood, and so this person would be sent up, as they said in New York, sent up the river. Okay, so STR analysis is very commonly used in doing these DNA profiling. So repetitive DNA, nucleotide sequences that are present in multiple copies in the genome. And these short tandem repeats, again, these are just short nucleotide sequences that will be repeated in random. So what you do is you compare the length of these STR sequences at specific sites, And you typically, to keep it low, as far as not, you don't want to get this exponentially difficult. Typically, you analyze only about 13 sites. And this is a lab that we would do, again, if we were face-to-face, is there is a non-coding region found in all humans that you would extract from your own DNA, you would extract from your own tissue, you would do it from cheek cells, and we would amplify it, and we would do a gel electrophoresis to show whether or not you had, how many copies of that short tandem repeat you had. And so that's what this means, is that some people can have 7 repeats, some people will have 8 repeats, and you can determine all of this using things like PCR. So you can have homozygotes, you could be a homozygote and have both alleles of that region are the same length. Or you can be heterozygote, that they can differ, one's a little shorter than the other, or you're just completely missing one copy and only have the other one. And you can determine all of that looking or using PCR and gel electrophoresis. And so again, this is just simply a way, this is just this particular marker here, AATG, this is just one of the ways that you can use to identify particularly something like a crime scene, is look at that person's short tandem repeats. And so here we're looking at, this is just a picture from another textbook here showing that we have a match here. We have the same length STR site. However, if we have 2 different samples and the site lengths are different, those STRs do not match. And this is how, again, you can determine whether or not a person may have been tied to a crime scene or not. You can also do this. I don't know how many people watch those shows like Maury Povich. I just saw it in passing the other day when I was going through the channels, that you could do these paternity tests with these. You can compare the STR sites between the parent and the child and see if there is some match between them. There's another more common way to do that, and I think it's in here. I think it's in this PowerPoint, so we'll talk about when we get there. But again, you can use this to look how related 2 people are. And so again, PCR makes exponential copies. You take the original DNA target region, you send it through the PCR process. You now have 4 copies. You send it back through the thermocycler PCR process again. And now in 32 cycles, after 32 cycles have been complete, you should have about 1.07 billion copies of that target DNA that are created. You can then do these, put these into a gel electrophoresis chamber. And what this will do now is through the, I'm trying to think of the best way to explain this without getting too complicated here. What this slide is showing here is that there are specialized polymerase chain reaction machines that you can say, you can put fluorescent dyes in the DNA, and if it shows up, you have a detector go, oh, you got it here. And in this region, you have this amount of DNA, or you have this amount of single the repeats in it, the STRs. And what you get is you get a printout that kind of looks like this. And so you can see the various different amounts of whatever short tandem repeat your heart desires, these regions that you're looking for, and how well or how not well the suspects, oh gosh, what are the, that's going way back in my brain, how well or not well the suspect's DNA matches those short tandem repeats. It has a specific name that just completely escaped my brain there. And so the probability of a random match is about 1 in 3 trillion, meaning that These are extremely precise ways to determine whether or not someone is guilty of a crime based on the evidence that you collect at that crime scene. And so the results can be obtained in less than 5 hours. That's a lot easier said than done because so many crimes are committed and so many samples are taken. Crime labs are backlogged for months and sometimes even years. The other thing, and that I always try to bring up with my students that has become a problem over the years is the bias of something like CSI type TV shows. They show that this can be just done in about 5 seconds. No, it's a very fast method. I mean, you can do it in 5 hours, yeah. But just dropping something into a machine, it's not going to automatically analyze it. That takes a human to sit there and analyze it to determine whether a person is guilty or not of a particular crime. And I call this the CSI bias because when you have juries now empaneled to hear a case of, let's say, a murder case, something very serious, they don't understand how long some of this stuff takes or the complexities of trying to understand or understand the data, because they're used to just seeing it on TV can be done in 5 minutes and it pulls up this picture of a person who it matches to perfectly and everybody goes home. That's not what happens. This takes a lot of experience and skill for someone to do the test, prepare the sample, do the test, and then analyze the results of that test. That slide back here, we already went over that, so that's why I just passed by it here. And I'm not going to talk about that one either. I'm just going to go straight to here. So this information here from these STRs here, these short tandem repeats, can be fed into a national system called the Combined DNA Index System. Now, when you do have all of this information from these tests, you can upload it to the FBI database. And so what this does now is that if more samples are taken from a crime scene at some time in the future, you can do the test and then you can compare it with what is already there. And what this does is you can link people to crimes, especially if it's someone like a serial killer who may be committing their crimes across multiple states, sort of like Ted Bundy did. So this was launched in October '98. It links all 50 states. What it uses is restriction fragment length polymorphisms. It can also look at 13 different single tandem repeat sections. And as we see here, there's a current backlog of over 1 million samples. This presentation is probably several years old now, so I would say that that's probably well over 1 million now of backlog. So what happens here, and I just stepped back a slide here, I wanted to talk about that one before we came back to here, is using these things called restriction fragment length polymorphisms, or RFLP. So what a restriction fragment length polymorphism, say that 3 times fast, is that if you want to determine the paternity of a child, what you can do is you can take a blood sample from the mother. You also take a blood sample from the child and from the alleged father. And what you do is you add a restriction fragment, the same restriction fragment, to all 3 members or all 3 blood samples. So what should happen is that if the mother and the child and the father, or especially if the mother is related to the child and the father is also related to the child should have a combination of DNA fragments that's a mixture of the mother and child, or mother and father. So that's what we're seeing here. So this mother here and alleged father here provided blood samples, and we can see their restriction fragment length polymorphism zones here. And we can see that the child has a 3 and 5 here. So after you've added these restriction enzymes and you let it sit and you let it make these cuts, you then can do gel electrophoresis and compare. Well, the mother has 5 and 7. The child has 3 and 5. But the father here, alleged father, has 4 and 7 here. That means that, let me see if this means that this person probably is not that child's father. Because the child is only showing, let's see, yeah, the child is only showing what the mother has here, a matching of 5. This is a terrible picture to talk about this, isn't it? But anyway, the results of the story are the same, is that if the father was the father of this child, we should see the child have some polymorphism that was similar to the father's. When you don't have that polymorphism that's similar to the father, then that person is not the daddy. And that's what they do on that Maury Povich show when he says, "You are not the father." That's what they're doing. They're doing paternity tests to determine whether or not that child has these RFLPs that are similar to both the mother and the father. If it's only similar to the mother, then the father is not going to be the father of that child. If it's a mixture of the two, then that person is going to be the child's father. Now, having also said all of that now, I don't know if RFLP is really used that much anymore, because now that PCR has become such a better and faster and cheaper method of doing gene amplifications, they probably use some PCR type tests now that will see if the same STRs are in the child that are in the father and the mother as well. Because to do an RFLP analysis, to do this type of analysis, you have to do some type of gel electrophoresis, and that would just simply take way too long. So they probably do some more automated thing now to determine paternity of the child. So going back just quickly here to gel electrophoresis here is, again, you have a mixture of different fragments and different sizes of the DNA. You put it in some electrical charge, and then after some time, you will see these bands start to form. And that's essentially what you're going to be doing. You're going to be taking M&Ms, I believe, and you're going to be taking the dye off of the M&M, and you're going to be loading that into your little gel electrophoresis and see whether or not the you're going to see the separation of those dyes in your little electrophoresis chamber. And so if you have something like a suspect here in STR analysis here, you can put DNA that was found at the crime scene, and you can have a sample of the suspect's DNA. And after gel electrophoresis has occurred, you can compare whether or not that person was at the crime scene. Well, the only thing that matches here is the short STR fragment. However, the longer STR fragment does not match between the crime scene DNA and the suspect's DNA. So this might be a little somewhat happier case for this suspect. This person was not at the crime scene, because to have been at the crime scene, it should have been a perfect match on the gel. You can do this to exonerate if you have more than one suspect. So again, you compare the crime scene DNA with suspect 1 and suspect 2. Okay, and so let me just get those out the way quickly. And so what we see is that this band matches this band over here. This one matches that band there. Okay, we got one match all the way across here. This band matches that, and then that last one matches that. Suspect 2 is clearly probably the person that committed the crime, and we can exonerate suspect 1. This is also just getting back to, again, that horrible picture before. You could also, this is kind of what an RFLP gel would look like if you were trying to determine if a baby was related to a father. And again, as we've said now, that this STR analysis, even though we have backlogs and even though we have better ability to do these kind of things now, we can exonerate people who have been in prison for many years. So just like Earl Washington here, I believe this is in the Campbell biology textbook. There's an insert about this because we talk about this in 1171. I believe the source for this was from a story on NPR a few years ago, but he was released in 2001 after 17 years in prison because finally his DNA was checked against the victim that he was who he had allegedly murdered. And it turned out that he was not the person who murdered the victim here. And it ended up being this person, Kenneth Tinsley, because the STRs matched semen found on the victim. And we can see here Earl Washington on the STR marker 1 did not match at all, or the marker 2, or even only a 12 on marker 3. So you can clearly see that Mr. Washington here was charged with a crime that he didn't commit. And again, a lot of this is also mired in socioeconomic and also racial issues. But again, we're not a sociology or political science class. So if you are taking those courses in your career, you can litigate those when you talk about that. But strictly from the genetic standpoint, we can use genetics and molecular biology to exonerate criminals from crimes that they simply didn't commit. So again, RFLPs can be used to detect differences in DNA sequences. We can have these things called, as we've mentioned, single nucleotide polymorphisms. This is just a variation at a single base pair within a genome. Again, restriction fragment length polymorphism is the change in length of restriction fragments due to the presence of some single nucleotide polymorphism that will alter the restriction site. And I just want to pull up the gel here. So when you're doing RFLPs, you can start to compare one sample to another simply based on how that restriction enzyme is going to interact with that particular single nucleotide polymorphism site. And again, you can use this to compare samples to see if a particular this DNA sample here has any relation to DNA sample 2. That's what you're looking for, is some type of relation. And this was used quite a bit during the World Trade Center tragedy of 9/11 in 2001. So just about 2,800 people were killed in New York City, and only about 1,650 of the 2,800 victims have been matched with their remains. And they're doing this through RFLP. They will take DNA that is collected from a victim of the World Trade Center, or even like the Pentagon was also attacked, and they will do RFLP on 2 different samples and then do gel electrophoresis to compare whether or not that sample belongs to a specific person. You can take it's just like, and forgive me if anyone has any relatives that were killed in this tragedy, but if remains were found and you were the closest relative to the remains found, and you had someone who died in this, you could give a blood sample and they could do RFLP on your blood sample and compare it to the sample that they have, and they could see relatedness between the two to tell if those remains did belong to someone that was a member of your family. And so 21,000 pieces of human remains have been collected. I believe this is still ongoing. They're still trying to identify all the remains they can. About 13,000 of those have matched up to DNA extracted from samples of victims provided by loved ones. Having come here to Ohio from New York, where I taught in New York, I tended to have a lot more students who had relatives die in 9/11 because, of course, it happened in New York. But this is still ongoing, and again, you can use these genetic analyses to figure out if a person is related to someone. Okay, so that's it for today. And I hope everyone has fun with that lab. I think over the summer we did it, and everyone tended to do very well with it and had a lot of fun with it. So if you have family members who like to do these kind of things, bring them in on it. Just tell them not to eat the M&Ms because that's your sample. So barring any questions, I will see everybody back next Monday.